Welcome, everybody. As Jeannie mentioned, we're really excited about this. This is, I believe, our fifth year of doing these webinars. And so just to kind of remind folks the purpose and intent behind these, many and most of the projects you're going to see today are collaborative partnerships we have with the Bureau of Land Management, the U.S. Fish and Wildlife Service, the National Park Service, and others. And so this is a way for us to provide updates on research funded through those coordination and collaboration. As Jeannie mentioned, today will be primarily focused on greater sage-grouse. Our last talk is a little bit about carbon. Every once in a while, we'll have a talk that gets sandwiched in for scheduling. But let's get it underway. Our first speaker, and if Pete, if you could begin to cue up your slides. Our first speaker is Dr. Pete Coates and Cali Weise from the USGS Western Ecological Research Center. And today they're going to be talking to us about the greater sage-grouse population monitoring framework and how we apply this range wide and talk about an early warning system for at-risk populations. So I can see Pete, I can see Cali. I don't see slides yet.
Looks good.
Okay, can you see the slides now?
Yes,
I can
thank you.
see the slides, I can hear your voice, I can see your face. Take it away, Pete.
Okay, excellent. Thank you, Leif. And yep, little bit of fumbling there, but okay. Well, I'd first like to say that I'm very pleased to be here, to be the first talk of the meeting here to launch the SURF series. I'm really excited about all the talks today. I want to say that I'll be tag teaming this presentation with Cali Weise. And I want to also acknowledge my co-authors in this talk, especially Brian Prohaska and Michael O'Donnell, responsible for a lot of the modeling you'll see, and the co-PI, Cameron Aldridge. I'm going to be presenting for you an update on the range-wide population trends and targeted annual warning system. And I'm going to just brief through a lot of this. We're going to spend most of the 10 minutes here, or a few minutes of the 10 minutes, focusing on the technical transfer toward the ending. This has been a colossal effort over the years and a massive collaboration across state and federal agencies. I want to thank so many folks, a few of them there listed in the left. The funding from the BLM was largely responsible for making this all happen, and all of the state agencies in sharing the data. Colorado State University for a lot of the logistics and modeling. Lots of folks here to acknowledge. I'm just going to brief through a few of the objectives here and talk about kind of what this framework's about at a really high level. I'm going to focus first on these two objectives, where we really focused years ago on bringing together a lot of data that's dates back 60 years and across the state's let count data to develop a unified range-wide population data set, so that we could monitor not just sage-grouse populations and the population dynamics, but also develop spatial clusters for various uses. When we look at the data set here, these are color-coded by state. The amount of LEC data, thousands of data points, as you could see from the numbers there that have gone into this, just a massive effort among the states and collaborating with them, those different state leads. This leads to a series of analyses that were done, spatial algorithms, clustering algorithms, and connectivity analyses to get to different population clusters that are nested. These are going to be used for various sources to identify population structure. Our main intention here is to understand trends across different spatial scales. And one scale of particular interest is the population level, a semi-closed population. And so we refer to this as kind of a finest scale, or neighborhood cluster, which we've identified this scale out of the 13. And then trends vary through time in a very similar way with climatic conditions at this much larger spatial scale, which we've identified the core scale here, the climate cluster. There's other spatial scales, population scales here that could be used for other purposes, but for the trends analysis and the targeted annual warning system, I'm going to focus on these two, one of which, again, is kind of the population level, and the other a much larger regional level. touch on the actual trend analysis here. When we look back at sage-grouse populations, or at least male peak counts through time, you see a lot of fluctuations, interannual variation, and it takes on a cyclical pattern about every 8 to 10 years or so we see these peaks and these valleys. And this largely is to do with precipitation. And this figure here, a 1-year lag effect, really demonstrates how tightly related these populations are in their cyclicity with precipitation patterns, temperature patterns, other types of climatic conditions. We have some new information that we're working on range-wide with that. This was a study conducted in the bi-state years ago. The reason why it's important to understand this is because when we're thinking about population trends, they could be very misleading based on start and stop locations of the time series. For example, if we start in this nadir or low point back in time, even oscillations back, and end on a peak, we can end up with a very misleading trend. Whereas we're really trying to focus on from nadir to nadir inferences so that we can align, for example, this trajectory of these oscillations with the trend. And the reason why we're using nadirs is largely because most biologically important is from a management standpoint, such that when populations are declining and we're trying to figure out extirpation probabilities, they'll using nadirs, they're as they decline to extirpation, we can make better predictions about when that might happen. And so when we look back at the range-wide data, we use a state space model to estimate apparent abundance through time. We could also estimate the slopes from nadir to nadir, which is the trends themselves. And we see 80% declines in sage-grouse population since the 1960s. This is approximately 3% decline per year. And again, thinking about it in terms of nadir to nadir is important, and across all these nadirs, we're estimating declines. But if we're looking at it on an annual basis, from 2022 to 2023, we had an increase from 3.7% increase. Now we're still working on 2024 data, so I don't have that for you today. We will have that the report published within about probably 2 months at this point. And have all that information. But this increase that we're seeing in the most recent year largely is to do with these oscillations. So this probably isn't a long-term trend that's changing, but just this annual variation that I talked about earlier. We're kind of seeing this same type of pattern across the entire range when we look at the different climate clusters. The more red the climate cluster, the steeper the decline. For example, the Great Basin there has about a 5% decline on average over the most recent nadir, which in this case dates back to 2013. Now we did see a 4.5% increase in that in the last year, but again, that just means we've come out of the nadir. The population is starting to increase a bit, largely due to the precipitation that we've had. We see this pattern across most of these other climate clusters too, with the exception of Washington, which had a decline of 15.4% in 2023, largely to do with those major fires that were up there, and really alarming for these populations. They might not be oscillating quite as much anymore and just in a decline. In the bi-state, we see a substantial increase of 19% in 2023 and one of the lowest declines since 2008, the last nadir at 1.2%. So if we look at the actual spatial variation through time, here these figures demonstrate this. We see more red out in this eastern portion of the range when these back in time there was a lot of cropland conversion and oil and gas development. But as we move to these more recent nadirs, we're seeing this shift to the western portion of the range, largely to do with invasive grasses, wildfire, things that we've really come to identify here as major threats to sagebrush ecosystems out west. Okay, so I'm going to move on to the targeted annual warning system. And what this essentially is we know from these long-term trends that populations are declining in most areas. Some neighborhood clusters are increasing, but for the most part at the climate cluster level. So we developed a system of which we can identify when populations at various scales, LEC and neighborhood cluster, have decoupled and are declining, decoupled from a larger spatial scale, the climate cluster. And this gives us an idea of those populations that are in trouble that we can manage on an annual basis and assign a watch or a warning and implement some type of of correction to them. So let's just take this climate cluster that's oscillating. What we would expect is the neighborhood cluster to be doing these similar oscillations with these precipitation effects. And it's typically around some carrying capacity that exists as they're overshooting it and undershooting it. And the population is regulating kind of around that carrying capacity, which carrying capacity is the number of birds the environment can support. But what is concerning is when we get populations like this that actually decouple from that larger spatial scale, largely because of some local perturbation like a wildfire or something else that's out there that causes them to decline and not necessarily respond to the precipitation like the larger spatial scale is doing. So we use two thresholds to get at these populations so we can identify them. One is the degree of decline and how long it's lasting, and the other is what that degree of decoupling is, signified by those orange arrows there. And the combination of this information can lead to some a pattern like this here, where we have no signal that results in a signal, then more decoupling and declining, a watch, and then this moves into a warning stage. Once they're recoupled, there's no signal, but it's still at a very low population and still of concern, which you could use other tools in the hierarchical framework to continue to monitor this population until it's essentially recoupled and back to its original abundance level. And so when we look at the TAWS for 2023, which is the warnings and watches across the LECs in the left figure, and the neighborhood clusters in the right figure, these are the populations that essentially have experienced aberrant declines. Those are declines that don't match this larger climate cluster, so they're not responding to the precipitation patterns of which they should, and there's likely some local perturbation there that's causing this. The last couple minutes here, we're going to focus on the software, and I'm going to turn this over to Cali. Cali, are you there?
Yep. Thanks, Pete.
Okay, so this is software focused on informing targeted, timely, and effective management. And I'll go ahead. Are you ready to play the video?
Yeah, go for it. So we're going to show an example of the software that we've put together that allows users to access the results of the TAWS and trends in their area of interest. So in this example, I'm going to bring in a fire perimeter. This is the Rush Fire that occurred in 2012. And so what I'm doing is by bringing in that perimeter, I've identified the LECs that fell within the perimeter of the Rush Fire. And you can see those displayed there in green. And we're going to actually pull up the results of the TAWS, the Targeted Annual Warning System, for these LECs, and looking at how the signals through time. So you can see we've got our LECs identified on this bottom map. We have a time slider that allows you to move through time to look at the signals that have occurred. So we're currently looking at the year 2011. That's 1 year before the fire. We can see we have a couple warnings in that population. The year of the fire, we have a warning before we're watches that occurred in the population. And then we can continue to move through time to see how many of these LECs end up with a signal, either a watch or a warning, most likely due to the occurrence of that wildfire. And so you can do this with disturbance, any kind of disturbance boundary, but you can also do it with treatment boundaries as well. So if you wanted to look at if some kind of management action had an effect on reversing signals, you could do that as well. And then we also produce this information in a table, so you can select any of the years of interest. So since the Rush Fire occurred in 2012, we're going to grab a year prior to that, and then about 5 years into the future. And this allows you to look at every LEC individually, and you can see whether a watch or a warning occurred. And that information is available. The map is available for download as an image, and then you can also download the table either as a CSV file or also as a shape file. And I do want to point out that we also include the trends results as well as extirpation probabilities. That's just in another section of the tool that unfortunately I didn't have time to show today. All right, next slide. All right, so we have several deliverables from this project. We have 9 published reports and papers and 14 data releases and software release products. Leaf just posted the link to our webpage in the chat, I believe. You can access any of the data the reports, the publications, and the software products through that link. Next slide. And then I just want to wrap up briefly talking about a technical transfer effort that we had. So this framework is very complex. We want to make sure it's accessible and as useful as possible to folks so that they can use it in their management decision making. So we formed a working group to identify some reference materials that we could produce for users of the framework, as well as developing a hands-on training module so that we could guide users through the framework and how to download, interpret, and apply the outputs of the framework. Next slide. So briefly, I just want to show some of the products that we have developed. So we have a project webpage, which the QR code is right there on the screen for you if you want to scan that. Again, also it's in a link in the chat. We have recorded some training videos that we are currently working to get through 508 compliance, and we'll have those posted online. That provides background on the framework. And then also we have developed in-person training module. We actually just completed our second training module yesterday in Colorado. So if you are interested in attending any of the future training modules or workshops on this framework, feel free to reach out to me. My email is at the bottom. And I can drop in the chat too. All right, thanks everyone.
So our second presenter today is also Dr. Pete Coates. And Pete is going to talk to us about how they used the aforementioned framework that we just discussed to talk about evaluating the effectiveness of conservation actions relative to responses in greater sage-grouse. So Pete, I can see your slides and the floor is yours.
Okay, great. Can you hear me as well?
I can hear your voice.
Okay, excellent. Thank you, Leif. Appreciate that. So yeah, as Leif mentioned, this next talk kind of segues right from the last talk where we're using the framework to evaluate conservation effectiveness of conservation or effectiveness of conservation actions rather across the range. I'd like to acknowledge the co-authors you see there, especially Brian Prohaska, responsible for a lot of the modeling that I'm going to show you here and some of the results. And again, the same collaborative effort with so many folks from the state and the federal agencies. This one was primarily funded by the U.S. Fish and Wildlife Service, but I do want to thank everybody, the folks you see in the left there, Colorado State University for the use of servers, et cetera, and all of our federal partners. It's been, again, a very large, successful collaboration. And as I showed in the last talk, populations have been declining. Back in time, you could see one thing I didn't point out in the last talk, but how these slopes and these declines, which are the different colors that you see there from these naders in the past, align so well. I mean, there's about a 3% decline on average. And the reason why this is important is because we're seeing similar declines in the most recent nadir as we have seen in some of the further out naders. And what this basically means is there has been more loss of sagebrush ecosystems, so we might expect these declines to actually be getting worse, but they're at least maintaining at some level. So that's kind of what this is about. Are we making a difference with conservation actions out there or not? And that I'm going to focus on that in this talk. And what we tend to do and what we have been doing in sagebrush ecosystems is more of an emergency care, if you will, type of approach, more of a reactive approach, and going to these areas that are most degraded to do conservation actions. There's been a recent conservation design strategy that's been published, you might be aware of, where the sagebrush ecosystem has been delineated into really 3 different spatial units, if you will, core area, growth opportunity area, and other rangeland. Core area being some of the most pristine sagebrush and most intact sagebrush area, and kind of outlines a design to protect those types of areas and maybe have some restoration actions with some of these other areas, especially the growth opportunity area, where there may be opportunity to grow the core. If we look at sage-grouse population trends within those 3 areas, we actually have stable populations within all of that blue region that you see as those core areas, which is there's no other actual type of boundary we've ever identified at that particular spatial scale where we have stable populations. So that was encouraging. And 22% declines in the growth opportunity, that's the light blue, and 64% in other rangelands. So maybe focusing efforts in the growth opportunity and reversing those declines is ideal. So this kind of gives us a spatial map of where to go. But the question is, have we been effective in the past? Well, fortunately, we have big data available to us now to measure this through the LTDL, Land Treatment Digital Library, and the Conservation Efforts Database, which are both very large and informative databases that bring in a lot of information across the range of actions that have been done over decades. 6 million hectares of core has been lost since 2000 to 2021, and many of the actions are aimed at drivers of that core loss. And so this gives us an opportunity to take these big data and combine them with our hierarchical model, which is other big data, which is all the state data on the response of sage-grouse, and look at a relationship between the two. And these are all the data points you see here of the conservation actions, over nearly 30,000 actions. 12,000 made the cut really for the use in the analysis based on some QA/QC work. I talked about the hierarchical modeling approach previously, but it's for those that didn't attend that, it's a state space model where we're counting for uncertainty and elect count. The difference here that we're doing now is we're using this information to do a progressive change before after control impact paired series study design. I'm just going to talk about what that means. It's a kind of the gold standard, if you will, in research, where we could control for any temporal effects that might be confounding our ability to look at it, something that a before and after type of effect. For example, here we have our impacted LECs, which are within 8 kilometers of each conservation action. And now we have control LECs we could look at within the same climate cluster and up to 150 kilometers to compare the trends from those conservation efforts individually to those control LECs through time. And we'd look at different lag effects, and we could also look at the influence of resilience, which is the ability for the landscape to recover from a disturbance, and resistance to invasive grasses. For example, in low areas, you have dry, warm soil, high or moderate R&R referred to as, we have moist or cooler sites. I'm going to show a series of figures from this point of some of our preliminary results, but in order to interpret the figures, we kind of need to go through this figure to show the interpretation. Everything's in terms of Bakke ratios. The ratio's there on the left, but what's most important to know is if you're below a ratio 1, you're underperforming as a treatment compared to the controls. If you're over a ratio 1, you're outperforming. Because this is a Bayesian analysis, we're working with probabilities, we get to really calculate the probability of outperformance to underperformance. Okay, jumping right to the results, immediate effects of conifer removal. In high R&R areas, we see increases in time in that outperformance, such that immediately after removing conifers, we have an effect, and it continues to increase through time. Whereas at low R&R, we still see a successful effect. You can see all those posterior distributions above the dashed line. It just doesn't get better through time. The effect just maintains outperforming every year compared to the controls. So both of them are encouraging. They're successful, but in high R&R, which tends to be in phase 1 areas, phase 2 areas where there's probably after the effect more understory growth coming back in, that might be responsible for why we're seeing an increase over time. We saw a strong effect, but not until 8 to 10 years on annual grass treatment, earlier for the high R&R, which we might expect compared to the low R&R. But how you interpret these Bakke ratios, I guess I'll start there too, so you can look at these the values here and make some sense out of them. This is about a 50% for any one of those years, a 50% outperformance from the control relative to the control, in looking at before to after on the population growth rate, and it's increasing 7.9% per year for high R&R and 6.8% for low R&R. And we really see these effects take place after about 10 years for annual grass here. Typically in these sites where annual grass has been removed either chemically or mechanically, they're typically at large spatial scale, sometimes following wildfire, which is where annual grass have invaded that area. And then there may be a sagebrush restoration action happening after that. We've looked at both seeding and planting of sagebrush on the landscape and across R&R. I'm going to kind of break this down. But we see about a 10-year effect here as well, which matches some of the research that we've done in the past, looking at sagebrush recovery rates in high R&R under a seeding scenario, 53% R ratio and a 9% increase. Here, though, in low R&R, we actually see the effect not really materialize until about 14 years, which makes sense because, again, this is a warmer, drier condition, but still successful on sage-grouse responses. Now, the average R value of 104, the reason why this might be more than the seeding in high R&R, caution here, and I have the years up there. This is 21 years of data. We didn't have 21 years of data in the high R&R. So you're going to have higher R ratio averaged values there. So that, might be an artifact of the years there as opposed to it being more successful. Just kind of a little caution in interpreting this. Planting had increases earlier, around 7 years in the high R&R, but took much longer, almost double that time before we saw any influence in low R&R. Now, the sample size, you can see the posterior distributions get very large here, and the sample size is kind of small there. So little caution too in that planting went out to 14 years in low R&R. It could have been one or two sites that we had a success at. All of these data that I'm presenting here are preliminary, and so we haven't published this work yet. So not for citation distribution, and just kind of exercise with caution as we go through, although we have come to completion on the modeling at this point, but just need to move through the review process. Okay, when we look at riparian restoration, an immediate effect in high R&R, but it stabilizes over time, whereas in low R&R, that effect actually, there's evidence that it increases over time. And this should be expected. In these low R&R conditions, there's less water and forb availability. It's a drier, warmer type of site. So it might having that restoration can lead to a much stronger effect, as you see from the average R ratio, where there may be more dispersed water and more forb concentration and availability across a landscape of high R&R. Okay, so that's what all I'm going to discuss as far as individual treatments. There's many more which will be published in the manuscript, but when we take all of this collectively, the average difference in R&R, if a conservation action happened, was 100%. That basically means that the population performance was twice that, the growth rate at the sites where conservation happened before versus after compared to control sites, and about a 10% increase annually at those sites. And so what we wanted to ask is, well, what does this mean for range-wide? Getting back to the original question that I posted, there were what I mentioned there about 3% declines. And so range-wide as a whole, when you take this into consideration with all of the control areas versus the treatment areas, it's about a 1% uplift, if you will. So in other words, these populations would likely, over the most recent nadir, be declining by 4% as opposed to a 3%. And this amounts to, over that amount of time since 2000, a 22% increase in abundance compared to no action. So this is encouraging. Conservation actions are working. And for the most part, they were guided by professional opinion. Some of them put in the exact spots, obviously, from the response of sage-grouse, but many of them may not have been. And having the conservation design strategy now available to us, we may be more precise and surgical in where they're placed in order to grow the core and also create a stronger uplift with less cost. I think the bottom line here is, though, that we still have a lot of work to do to reverse these declines that are still happening at around 3%. What we showed here is there's a lot of variation in effectiveness of these actions, and the more information we have, the more we'll understand that variation and get more precise and surgical about the type of action that's going to make the most difference. It fits well with the sagebrush conservation design strategy and the hierarchical framework in order to focus on these growth potential areas and grow the core. And as we mentioned in the last talk, the targeted annual warning system points out where populations are in trouble for sage-grouse. So having information like this, and then ultimately being able to monitor it through the hierarchical framework, monitor the outcomes of that action when it comes to population response of sage-grouse, would be ideal. And we're planning to incorporate that into the hierarchical tool that Cali presented on in the previous talk. how you could just put in your restoration site, for example, and derive the types of figures that I just showed here at those specific projects. We have a few deliverables at this point. Most talks and some preliminary data that was published to show that this system worked at a smaller area, the bi-state population, but we will be having a couple manuscripts and data releases, et cetera, come out this year on what I presented on today. And thank you very much. I'll take any questions if I have time.
So thank you again, Pete. Question from Gregor and John in the chat for you. Okay. Our next speaker is Dr. Greg Wann from the USGS Fort Collins Science Center. And today, Greg is going to be talking to us about a few projects, primarily centered around mapping of seasonal habitats for greater sage-grouse and identifying differences regionally in the thresholds and seasonal habitat use. So, Greg, I can see your slides.
So yeah, thanks for the introduction. And yeah, I'm going to be covering a couple of different projects, but they're really all derivative from our range-wide seasonal habitat mapping project. So I want to start off by acknowledging co-authors on this. We have a really big group of people that I'd like to say thanks to. So first of all, our funding agencies, Bureau of Land Management and USGS provided funding support for this. Very special thanks to Ashley Whipple for all the work that she did compiling the spatial data sets. Thanks to our core team group that's been with us from the beginning on this project to help develop the models. And then a very, very long list of data contributors who shared their hard-earned telemetry data set so we could create these models.
Greg, I'm going to interrupt you for just a second. We can't see you on your camera. If that's by design, that's fine. I just wanted to let you know that your camera's off.
I can, 1 second.
Okay.
So I'm trying to find the screen. So is it okay if I just try to flip that back on at the end of this?
Yeah, that's fine.
Okay.
That's fine.
Yeah, so to start off, just an outline on this, I'm going to provide some information on the motivation for the project. I think many people on the call have probably seen our at least the 1 or 2 presentations that we've given on seasonal habitat models. I'm going to go through that one pretty quickly. And then that way we can talk about the 2 funded projects for creating discrete habitat mapping as kind of derivative from those seasonal habitat models. And then another project that's an offshoot, which we have just started, and that's basically using the seasonal habitat data sets as predictor variables in a growth modeling exercise. I will talk about some other extensions for this, and then finally go into the timeline for those those last 2 projects, projects 2 and 3. Okay, so to start, the motivating factor for this project was really to help the Bureau of Land Management with their evaluations of greater sage-grouse seasonal habitats. So as you can see from this map on the right here, the Bureau of Land Management basically has land that they manage occupied by sage-grouse across the range-wide extent. So they manage about 50% of that land. So they've got a pretty big responsibility in land stewardship. And one of the ways that they do the assessments of these habitats is through a document called the Habitat Assessment Framework. And the Habitat Assessment Framework, it's a very large document and it covers a lot of aspects of habitat evaluations. But I just want to focus on what we're actually doing, and that's the component of the HAF that basically focuses on evaluating seasonal habitats. Okay, so there are a lot of different products available out there to evaluate seasonal habitats. A lot of map products, including some products that exist at the range-wide occupied range. So this is an example of a probability of extirpation layer produced here by Aldridge et al. 2008. This is a type of product that doesn't specifically get at seasonal habitat models. It's out there. We also have other types of data sets that are really, really good for this exact purpose. So here's an example by Walker et al. Of some seasonal habitat maps produced in Colorado down in the Piceance Basin. This is restricted to that region, so they're not range wide. And there could be some issues with comparability whenever you have these types of maps too, because they might define seasons in slightly different ways. have different predictor variables that go into them, and they're summarized different scales and stuff. And then the meaning of the actual pixels can vary too. So here's a cool example that people have recently really started to do, which is basically deriving layers that look at habitat selection, but also things like demographic rates to identify source sink habitats. So obviously, pixel value in the map on the right there is going to be comparable to a probability of selection like the one in the middle. So basically, this is trying to create just a consistent, continuous map across the sage-grouse range that's specifically reflecting seasonal habitats for sage-grouse. So presumably these are important types of studies that are being conducted and must be well used and funded, because we're seeing a really big increase in the number. So this is a plot that shows a Google Scholar search for the terms resource selection and habitat selection controlled for the total number of sage-grouse studies returned for each year. So obviously that's going up a lot. When we first started this project, we wanted to really try to understand, what's already been done. So we conducted a lit review, and this plot right here shows some results from resource selection function coefficients and the proportion of those coefficients that were positive for different types of environmental predictors. And then we've got this summarized here for across all seasons, and then just broken down separately for spring, summer, and winter. And kind of one of the interesting things that you can see here is that we have some consistently avoided types of habitats that are being reported across, I think this was about 56 studies that are summarized here. So, there's some variables that you consistently see avoidance for. We also see that there's some other types of habitat conditions that are consistently selected. But kind of here in this middle range, we've got all these that are mixed. So a lot of studies are reporting that there's selection for these features. Other ones are, reporting avoidance. And I think that can create some difficulties in interpretation, from a management perspective when you're trying to do these syntheses and understand what's going on. So I'm going to be pretty quick with the seasonal habitat models in terms of methods. So yeah, we compiled a really big telemetry database from a bunch of data sharers. We really appreciate their telemetry data sets to let us do this. And we fit resource selection functions using conditional logistic regression and lasso. And then basically used tenfold cross-validation to optimize our model. And then we also did a robust validation procedure. So we withheld a subset of data sets from different study areas. So these are actual, spatial withholds. And at the end of the modeling, we confronted our model with those to see how well they were able to predict out of sample data. So I think this plot here kind of summarizes resource selection functions really nicely in the way that we're kind of thinking about this and the setup of our data. So these are 8 different panels here. They show different regions across the sage-grouse occupied range. And then within each one of these panel plots right here, we have the 6 different seasons that we modeled. So we've got nesting, early brood, late brood, breeding, summer, fall, and winter. And then we have an available distribution and a used distribution. So these are paired for each of these. And we can kind of see how the distributions for one example covariate, which is sagebrush cover, varies. And, if we look, we can see that almost always sagebrush is used more than what it is available. And if we kind of look across regions, we can see that this varies too. So for example, for the nesting season right here, the magnitude of the difference between these 2 distributions is quite a bit bigger down here in the bi-state than it is over here in Montana and Dakotas. Similarly, we could take any one of these seasons and make some comparisons here, and we see, for example, the late brood rearing season. There's not much differentiation between these 2 distributions compared to like the nesting season. So that indicates that basically we do have some regional variation here that we should probably at least test for, and the seasonal periods can make a difference as well. So there might be different selection patterns by season. This is a map of the range-wide greater sage-grouse distribution. And we use the O'Donnell 2022 cluster levels that Pete Coates talked about in his presentations. And what we did is we used those to assign the different telemetry locations to regions and sites. So we kind of have this cluster hierarchy in the modeling that we wanted to account for. So we used the level 12 clusters, I believe it was, in the O'Donnell data set. And so each one of these different colored polygons represents a different region. And then nested within those regions, we also have site level polygons. And then here's an overlay of the telemetry location. So each one of these points here shows the median XY coordinates of all the different sage-grouse that exist in the data set, the range-wide data set. The purple locations are the ones that were used in model development and training. And then the red locations are birds that were assigned to randomly selected sites for model validation. So one of the things to notice here is we do have, I'll just go back one slide here. So we do have telemetry locations represented in all the regions, but a lot of the sites, we actually don't have telemetry locations. So these are all the gray polygons here. So this model does do quite a bit of extrapolation. Okay, so for the predictor data set, we had a lot of candidates, and they needed to cover the range-wide US occupied sage-grouse range because we were making maps. And they included things like annual land cover, covariates, disturbance features, topographic attributes. And then, we calculated or measured these at use in available locations using either moving windows or distances. And we did do a scale optimization. So we attempted to identify the optimal scale for each of these covariates. So it was a pretty big predictor data set. I think we had about 30 total predictors. And then we also had, different measurements of either moving windows or distances that we did this for. And those are all candidates in the models. Okay, so we created predictive models for 6 different periods. And those included like life history periods. So we had a nesting period, early brood, late brood, and then we had general seasonal models. So those were created for the spring, summer/fall, and winter period. And we did predict our models across the occupied sage-grouse range. So our predicted maps represented relative probabilities of selection that range between, so they were scaled range between 0 and 1. And these scaled RSF scores are basically the core data sets produced from the seasonal habitat modeling project. Okay, so one of the things that's tough, I think, from a management perspective, is to take these continuous RSF maps and, how do you, the question really becomes, so how do you know if the probability value is high enough to really consider that to be habitat or not? So that gets into the kind of the working part of this, which is figuring out how to go about doing thresholds for this. So on the left-hand side, here's an example of our winter map. This is on the continuous RSF score, so relative probabilities of selection. Those black triangles are LEC locations. And then on the right side is doing some thresholding procedures here to basically discretize those RSF probability scales into different categories. Okay, so that brings us to our first extension of the project, which is to focus on tech transfer. So how do we work with continuous seasonal habitat maps? So this is ongoing work. We've done some thresholding basically to hand off maps for review to managers and other people involved in this project. But this is one of those places where we do need some more input from biologists in terms of understanding what their specific information or, the specific goals that they have for use of these habitat maps actually is, so we can better optimize the thresholding procedures. Okay, so here's an example of, So here are like 2 different examples of how we can go about doing these thresholds. So one is to basically use bin cutoffs at targeted RSF quantiles. So those quantiles would be at the use locations of the telemetry points. And then we can do something like maybe doing, a non-habitat all the way up to high quality habitat types of bins. So this is a pretty common approach to use to discretize continuous probabilities. And then there's also another type of optimization that could be done where if you just want to, give a binary yes, no, it's habitat, it's not habitat. And that's to identify the cutoff that basically captures the most use locations while implicating the least amount of area. So that's another way that we did it too. And this is a method used by Fetty et al. 2014 in their monograph on seasonal habitats in Wyoming. So we have done both of these, and I can show you an example of this last one for optimization here. So this is a table that shows different thresholding scenarios in terms of the percentage of sage-grouse locations captured in the landscape area implicated for our winter model. So here we identified optimal cutoffs separately for each of these 8 different regions. And there's some variation across the range. So in the bi-state, we're able to capture about 90% of locations and 25% of habitat. We could also look at eastern Wyoming and Colorado here, and we see that that same amount of landscape is only capturing about 70%, so it does vary. We can also just hold those criteria constant, so the landscape required to capture 50% or 85% respectively here. And we can see that there is quite a bit of variation across these different regions and how we do that. So if we take the average across these regions, we can see that it takes about 31% of the landscape to capture all the use locations here. That's whenever we do these regional specific thresholds. We just performed a single unitary threshold across the range. We capture about the same number of birds, but it would actually be 40% of the landscape required to do that. So we do gain something from doing regional optimizations here. Okay, so the last derivative project or extension here is, we haven't done that. We actually just started this recently, but the basic goal is to identify contributions of seasonal habitat quality captured in the RSF scores and greater sage-grouse population health captured via growth rates. So the plan is to basically use the RSF scores to evaluate contributions of habitat quality to variation in growth rates captured in the TRINS data set that the group out in California with the Pete Coates leading is done. And the outcome there would be this tool to identify which seasonal habitats have the most potential to improve population health or which should be prioritized for continued management. So this is an example of a derivative assessment that we worked on last fall using the seasonal habitat maps that was kind of fun. So we basically what we did here is we used the annually timestamped data sets to make back-in-time predictions of seasonal habitats from 1995 to 2020. So these tables on the left show our calculations of the percent change in the continuous RSF scores in the discrete binary habitat maps for the general seasons of the spring, summer, fall period, and winter. So down at the bottom in each of these tables in light gray here, we've basically also calculated a composite annual map derived from the individual seasonal maps. And we calculated that in two different ways. I'll just focus on the multiplicative. And we've got the changes in habitat, which is obviously mostly loss here from 1995 to 2020 for these two different types of maps, either the discrete or the continuous. Okay, so the map on the right here, the cream colored plots are showing areas that's basically neutral or no change. Red is loss, and then blue is gain. So most of the predicted habitat loss here is located in the western portion of the range. And if we use the continuous RSF scores, we're calculating that there is more habitat loss overall. So what are some of the drivers of that loss? So this plot here shows an example of how one of the important predictors, sagebrush cover, drove changes in habitat from 1995 to 2020. So this is based on a sample grid of 1 million points spread across the sage-grouse range-wide maps for 3 different seasons, and how the percent change in the RSF scores relates to the corresponding change in sagebrush cover. So decreases in RSF scores are primarily attributed to losses of sagebrush, so that explains the most variation in changes in the map, but it doesn't explain all of it. So there are also a lot of other important drivers included I'm not showing here, and those include things like increases in annual herbaceous cover and the amount of burned area. Okay, in terms of product implementation, tech transfer, we haven't started this yet, but we do plan this spring and hopefully through the summer to be working closely with BLM biologists to take those continuous RSF maps and then try to make some customized habitat categories for them once we really understand kind of what their use is going to be with these maps. I mean, there are a lot of different applications for these maps, but that'll kind of help us target the best strategy to go about making that continuous map discrete. And then we're also going to begin that process of evaluating the population trends as a function of the seasonal habitats to see if we can sort of identify if there are any specific seasonal habitats that are explaining the most variation in growth. And obviously, habitat doesn't explain everything about growth. There can also be other things outside of that the habitat maps wouldn't be covering. But from a habitat management perspective, this is, I think, a pretty interesting type of analysis to do. And then potentially completing our back-in-time mapping. We had not, well, we've obviously done some of this stuff and initiated it, but that's not yet finished. And I do think there's some pretty good potential for some app development for this as well, particularly for the optimization in terms of seasonal habitats and growth rates. We do have some peer-reviewed publications out or about to be out on this, and then we have several that are in the works right now. So say if you have any questions about this, feel free to reach out to us, either myself or Cam Aldridge. Our emails are here. And there's also a website here where you can find some more information about the project. So thank you.
Thank you, Greg. All right. Our next speaker is doing back-to-back talks. So I'll just introduce her once. We'll pause in between for questions. But Dr. Shawna Zimmerman from our USGS Fort Collins Science Center is going to be talking to us initially about some sage-grouse genetics and characterizing the environmental drivers of range-wide gene flow. So Shawna, I saw your face. I can see your slides in presentation mode.
All right. Wait, you see the presentation part or the actual just slide?
Your slides look like they should.
Okay, perfect. All I needed to know. Okay, so this project is funded by the BLM Wildlife Program, and I can't remember if we've said this or not, but this is a New Start project, so I'm just going to be introducing it today. So when we think about movement among populations on a landscape, it should be relatively frequent and easy across high quality habitat when there aren't a lot of barriers. And so this should result in relatively high amounts of shared genetic variation or high gene flow range-wide. But we know that landscape composition is often complex and the different features. Okay, so I will pick back up here. So we're talking about complex landscapes and gene flow patterns. We expect that gene flow patterns and how we see that show up in genetic variation patterns will be differentially influenced by the composition of the landscape and the degree to which the different features actually affect the gene flow process. So these patterns of gene flow actually represent more contemporary impacts to populations, and they're considered good proxies for understanding our expectation of adaptive ability to future conditions, the likelihood of avoiding things like inbreeding depression, and just generally resilience and health of populations. And so having an understanding of these relationships between gene flow and the landscape can be useful for identifying which populations are at risk, which features might be kind of the primary culprits of that, and which actions might be most appropriate. So this is information that can be used in management planning and implementation of specific actions. So the specific project is to use landscape genetic connectivity modeling to characterize drivers of gene flow across the greater sage-grouse distribution, and then create a predictive gene flow surface. So our modeling approach is going to use this idea of the landscape as a resistance surface where different cover types are variably permeable, which allows us to model gene flow on a landscape as analogous to electricity moving through a grid. So in circuit theory, we have electricity that moves between nodes in a grid as moderated by, or at a rate that's moderated by one of these physical resistors, this squiggly thing in the diagram. And when we think about gene flow on a landscape, we have genetic variation that's passed between populations at a rate that's moderated by the composition of the intervening landscape. So the more shared genetic variation, the more easily permeable the landscape, or the more permeable the landscape is. So we're going to fit a model that was developed in 2013. I'm going to symbolize it like this. It models the genetic distance that we observe between points on the landscape as a function of the specific landscape features that will be in raster form and the degree of influence that each of those features has on the gene flow process. And this piece, this degree of influence, we're actually going to estimate in the modeling process. And I point that out because it's a significant improvement in the modeling, landscape genetic connectivity modeling, where we've historically had to rely on expert opinion to inform this piece in the modeling process. So now we can let the data tell us. So we have range-wide genetic data from previous projects, and we're going to work with some of the spatial data that Greg just talked about as part of the seasonal habitat modeling effort. And we'll fit multiple combinations of models with those different spatial variables so that we can identify the best fit version and then use that to create that predictive gene flow surface. And so the things that we can take away from this that can be useful, those expected deliverables, are essentially a ranking of those best fit models, which can tell us about which features are the primary drivers of gene flow and how much influence they actually have on that process. And then the predictive gene flow surface can be summarized in various ways to inform management planning and the implementation of different actions. So for some potential applications, I'm going to rely on some previous work we did for the Gunnison sage-grouse as examples. So we used a really similar modeling approach for the Gunnison sage-grouse, and we modeled gene flow at 2 scales. And I'm going to focus on the basin-wide scale, where we modeled gene flow between the leks within the largest Gunnison Basin population to illustrate this, the applicability. So this is a table just of the top-ranked basin-wide models of gene flow. And so when we look at this, we can think about which of these variables are showing up in our top-ranked models consistently, and we can look at the coefficients associated with the different variables and get a sense of how important are they. So larger magnitude values are more influential, and then in which way are they influential. So positive values are facilitators of gene flow, while negative values are inhibitors. So for the Gunnison sage-grouse, we effectively found that the height of the shrub community, which is almost entirely sagebrush cover here, was the most important predictor of gene flow. So taller shrubs were associated with higher gene flow. And this was accompanied in our best fit models by other modifiers or descriptors of that sagebrush community. So overall, it mostly affirms that the complexity and the composition of that sagebrush community is an important part of gene flow for this species, which is not surprising. So for the gene flow surface, areas of high gene flow are generally expected to be healthier and more genetically resilient. And so those might be good targets for protection or preservation. While those areas of more moderate gene flow, those might be best suited for some of the more active management actions like restoration. And then if we're thinking about the low gene flow areas, you know, the context might help us decide what those might be best suited for. So if it's a peripheral area, then it might just be naturally low because it has fewer neighbors to exchange genes with. But if it's an area kind of sandwiched between relatively higher gene flow, then that might be an additional opportunity for some of those active interventions like habitat improvements or restoration, or maybe even translocations. Similarly, if we're thinking about like the sagebrush conservation design that you heard Pete talk a little bit about earlier or introduce, those areas of high and moderate gene flow might be considered part of defending and growing the core of sagebrush habitat. And within the TAWS or the genetic warning system, these data can also be summarized within those, you know, population clusters and give us a little more context about what we're seeing in the population trend or the genetic diversity. So this last example is about using simulation of gene flow across these predicted gene flow surfaces to predict how we expect gene flow patterns to be affected by changes on the landscape. So for the Gunnison sage-grouse, we took our gene flow surface and created different versions of it, where we identified 2 leks with kind of moderate levels of gene flow and modified that shrub height component to either increase the shrub height, expecting because of that positive relationship we found, that would improve gene flow and result in fewer genetic differences between the lek over time. And then we also created a version of that gene flow surface with the opposite. So we decreased the shrub height, expecting less gene flow, and that would result in more genetic differences over time. I know this is a little confusing with the increase decrease, so we thought about it as a habitat improvement, where we would expect that to result in less genetic difference over time, and a degradation where we would expect more genetic difference over time. And then when we use the simulation or do the simulations of gene flow across these different gene flow surfaces, we actually found the patterns we expected. The habitat improvement resulted in fewer differences in genetics between the 2 leks, while the degradation resulted in more. They took different amounts of time to start showing up in our simulations, but these patterns were as a result of that landscape modification and changes in those gene flow patterns. So these are just some of the examples that could be where these kinds of analyses could be useful. So that's what I have, and I'm happy to take questions or comments.
Wrap that, and let's move on to your next presentation. So Shawna's next project that she's going to talk to us about is characterizing greater sage-grouse climate-driven maladaptation. And I can see your slides. I can see your face. We just heard your voice. The floor is yours.
Okay, thanks, Leaf. So this project again was all funding is from the BLM Wildlife Program, and it is again a new start. So I'm just talking about concepts and introducing where we're going with the project. So we know that environments can shape populations over time. So for example, populations where some individuals have traits that allow them to better cope with heat in a hot climate, those individuals are likely to be more fit in that environment. And so they might be more likely to survive or reproduce more frequently or easily. And so they're going to pass on more of their genes to the next generation. So over time, we're going to expect that the traits they have, as well as the genes that underpin those traits, are going to become more common in that population. And so you're probably not surprised. This is a very simplified version of natural selection. I'm sure you all are aware. And we know that environments are really complex, and so are the mechanisms or traits that populations have that make them best suited for a particular climate. So these patterns of adaptive divergence are an important part of understanding the basic biology of a species, but they're also really important when we think about managing declining species in a changing climate. And those are things that we think about for the greater sage-grouse. So as the local climate changes, populations may find themselves with traits and genes that underpin those traits that are no longer suited for their local climate. So this is the idea of maladaptation. So the greater the shift in that climate, the greater the risk of maladaptation. And this is information that can help us understand, again, which of these populations are most at risk from climate change, and inform management planning and the implementation of specific actions. So our goal is to understand how the greater sage-grouse is currently adapted to the local climate, and then to predict how suitable the climate is expected to be for the greater sage-grouse in the future. So we're going to use genotype environment association analyses to understand these relationships. So we have whole genome sequence data from across the species distribution. These represent sites that cover the range of variability in the climate. And we're going to work with bioclim data. So at their core, these genotype environment association analyses, or GEAs, are about finding strong associations between genomic variation and environmental variation. And those environmental variables are considered potential drivers of natural selection. So in this case, we're focusing just on climate. And once we've characterized the relationship between the genomic variation and the climate, we can use this to create spatial predictions of adaptive similarity. So similarly colored parts of this, dummy data distribution here indicate similar relationships between the genetics and the climate. And so we can then swap out the current climate variables with climate change predictions and redo those analyses and estimate what's known as the genomic offset. So this is the amount of genetic change that it would be required to keep pace with the predicted change in climate. And the genomic offset can be thought of essentially as the risk of maladaptation under climate change. And then we can also create spatial prediction layers of that genomic offset, and this represents the distribution of maladaptation risk. So for deliverables, we plan to create raster predictions of adaptive similarity, describing how greater sage-grouse are likely adapted to their local climate now, and then how that is expected to change in the future, and then also those spatial predictions of the genomic offset under climate change scenarios. So these are data that can be summarized in a variety of ways to guide management planning and implementation. So the most direct application could be to simply rank and identify threatened populations with that genomic offset or that risk of maladaptation. So here I've identified some populations that are of low, mid, and high risk of maladaptation. So these could be used to prioritize how limited resources might be used, or just to understand which areas are most at risk. So we're using these just generic circles here just for illustration purposes. But when we actually have data from the greater sage-grouse, as with any raster, any delineation can be used. It could be a population boundary, any useful management boundary, or the hierarchical population clusters of the TAWS and the GWS. Within a resist, accept, direct framework, or that RAD framework, the areas of lowest risk might be good targets for conserving or protecting. Areas of moderate risk might be the best targets for active management interventions, while those highest risk areas might be those that are kind of really costly to focus on for long-term persistence of the species. We can also use these predictions of current and future climate suitability to inform different management actions. So if the goal is to think about augmentations or reintroductions in the present, that current adaptive similarity data can be used to identify suitable source and recipient populations. If we're thinking about future proofing or being proactive about the effects of climate change, we might base selecting those source and recipient population matches for translocations or reintroductions that we're implementing now based on matches to the future adaptive similarity. And if we know that there are areas that are predicted to have large shifts in that climate suitability and we'd like to preserve them, then implementing actions that might facilitate natural range shifts or active shifts in the range into the better suited future climate might be reasonable options. So again, this is just a brief overview, and those are just some examples of how this can be useful. And I'll take any questions if you have them.
Is going to be talking to us today about quantifying carbon storage and greenhouse gas emissions in sagebrush rangelands to inform management of carbon resilience. Seren, I saw your face. I see your slides.
Awesome.
And
Great.
I hear your voice. The floor is yours.
Great. Thanks for putting me in today. I'm excited to talk to you all today about something a little different than we've been focusing on, move below ground a bit, and think a little bit about the effects of fuel reduction treatments on rangeland soil organic carbon. So this work is part of the Sagebrush STEP treatment evaluation project, and funding for this individual work comes from the Fish and Wildlife Service and the Northwest Cask. So sort of getting right into it, there's a growing interest in rangeland's ability to sequester and store soil carbon, which is associated with several ecosystem services like soil, water, and nutrient retention. And that interest outpaces our knowledge of rangeland soil carbon dynamics, which can be pretty tricky to understand. SageSTEP is a long-term, large-scale experiment that aims to provide managers with the best info possible to make decisions with reduced risk and uncertainty, sort of making it an ideal place to study soil carbon dynamics and move this needle forward. But before diving into project specifics, I thought it could be useful to just give sort of a brief generalized overview of soil organic carbon and how it comes to be. So soil organic carbon, which I might shorten to soil carbon throughout this talk, is found in soil organic matter. And it is the result of the balance of our carbon inputs into soils, so these green arrows driven by plant activity, and outputs, orange arrow out of the soil driven by microbes in the soil. So plants add carbon to soil by dropping litter and through root activity, and then microbes are decomposing some of that carbon back out into the atmosphere. And like many ecosystem processes in drylands, soil carbon change can be pretty slow, but there are some drivers of change that can impact soil carbon storage on management relevant timescales. So one really important consideration is vegetation, your plant community, how it's changing, because that can impact what we call plant soil feedbacks. And so I like this diagram because it shows the different rooting structures. You can sort of compare between some native species. We've got a sagebrush here, cheatgrass, and then even thinking about the interspaces as well. And just sort of highlighting that a lot of plant carbon inputs come from below ground sources, come from root matter and root activity. These differences in plants can be pretty important in terms of how much carbon is entering below ground. Climate's also really important. Temperature and moisture regimes influence soil conditions, which might impact microbial decomposition rates, but also feedback into plants and the types of plants that can grow there and how they're growing. Fire and shifts in fire activity are meaningful for soils. They can have a direct impact on soils through combustion of soil organic matter along the soil surface. And then again, feedback into changes in vegetation. And management, which also, again, is sort of a direct disturbance to soils, but may also lead to changes in plants. So each important and each considered in the study, but all sort of a conduit, or vegetation is kind of the conduit for a lot of them in many cases. And today, I'm going to be talking a bit about a sort of unknown within the management realm of this, which is the impact of fuel reduction treatments, while also considering plants, climate, and other things that might be impacting carbon. So I asked this question using data from the Sage-Step plot network, which is this large-scale study covering much of the Intermountain West. And at each of these sites that you can see, they fall into one of two categories, a shrubland site or a woodland site. So there's kind of these two networks across the whole plot network. Both sagebrush ecosystems, the woodland ones experiencing some phase of conifer encroachment. And each of those sites, there were 4 fuel treatments applied. So importantly, each had a control, and then some, there's for fire, mowing, application of an herbicide in the shrubland plots, fire, mastication at a subset of plots, and cut and drop. And then at each treatment in each site, there were several subplots where 190 centimeter soil core was taken at 15 centimeter increments. And those soil cores were taken prior to and 10 years after treatment. And so that sets us up with all the data that I've had to play with to kind of address this question. So in terms of what we expected to find, we thought that treatment would likely lead to an overall decline in soil carbon, largely because we've removed a lot of the plants and thus all of our green arrows of carbon into the soil. So we thought there would be an overall decline. Because soil change likely happens pretty uniformly throughout the profile, change often varies by depth. We expected there would be some depth-dependent change. And also, given the really large range that the Sage-Step network covers, we thought that environmental variation would also be sort of meaningful in identifying treatment effects. So sort of going through those one by one, this kind of gives us a coarse overlook of what the data looks like. So we're looking at soil organic carbon change here over 10 years in each network, colored by the different treatment. And you can sort of guess, just probably by looking at this, that there were not really significant treatment effects on soil organic carbon. So the gray boxes are controls, and then they don't look all too different when we're comparing to the different colors representative of the different treatments. So no declines. And in fact, this soil organic carbon actually sort of increased across the board in the shrubland network, which was interesting. So we rejected our first hypothesis. Next, if I just break that data up by depth, so instead of looking at the full 90 centimeter core, breaking it all up by the 15 centimeter increments, there was one depth-dependent effect, which was that in shallow soils, there was more soil carbon loss in the low treatment compared to other treatments. But aside from that, again, probably pretty visually apparent looking at this plot, not a lot of change. And so we give this one sort of a soft reject with the one exception. And then finally, what happens, does where you are on a map matter in terms of treatment effect? We found that including environmental variation did improve model fit, but did not lead to any treatment effects kind of popping out. So sort of a yes and no here. Soil carbon change was partially explained by environmental variability, but not in a way that was tied to treatment type. So some takeaways here. Treatment effects were limited, but there's a really sort of important but to this, which is that this finding likely does not scale to account for spatial heterogeneity. So while our study covers a really large geographic region, the coarser scale of soil sampling likely doesn't pick up on these sort of microsite differences that we might expect under different types of plant cover. So just something to sort of keep in mind. And also because we know that with time, prolonged vegetation change can impact carbon, it's possible that additional changes will become apparent over longer timescales than the 10 years that we were looking at here. So with not a lot of treatment effects popping out, I did have sort of one more question that came out of this, which was what was driving soil carbon change, if not treatment type, particularly in the shrubland network, where we were seeing a pretty consistent increase over this 10 years. So looking beyond treatment type, what was predictive of this change in shrublands, of all of these kind of things that we were considering? We found that the greatest carbon increases, to name a few, there was more in this list, but to just highlight some, were occurring in areas that might have been lower elevation, had less precipitation, and less perennial grass cover at year 0, so pre-treatment. And this is really interesting because these are all sort of low productivity conditions that we wouldn't really expect to match with an increase in carbon storage. But importantly, another variable that was related to carbon increase was an increasing perennial grass and forb cover over the 10 years. So thinking of grass change as change instead of just that year 0 condition, which is super interesting and kind of tells us that low productivity sites may have the capacity to gain soil carbon if they are also gaining perennial grass and forb cover. And also kind of speaks to the importance of plant-soil interactions, even just in this 10-year time frame. So really quickly wrapping up, field treatments had little to no effect on soil carbon, but we expect that this would vary with space and time. Treatment type aside, low productivity shrublands have the capacity to gain soil carbon, particularly if they are gaining some perennial herbaceous vegetation. We've got a manuscript in prep for this that we hopefully should be complete relatively soon, and some upcoming work. The next step is taking a look at the ecosystem level carbon change across this 10 years, so considering all of our above ground components as well, and feeding our soil carbon data into a larger soil carbon Great Basin synthesis. So yeah, thanks, and take questions if there's time.
All of our speakers today, Pete and Cali and Pete again, and Greg and Shawna twice and Seren. So this is great. As we mentioned, this is our first try at the 90 minutes. We made it in under the wire, although I'll continue to talk here for another 60 seconds, I'm sure. I had a former supervisor tell me that feedback is a gift. Doesn't always include a gift receipt, but we'd like your feedback on how this went. There's some tweaks and things we might be able to adjust between this session and the subsequent 4 sessions. So with that, Jeannie, I don't know if you have anything else that you want to share to close us out.